Next up is Michael Zemel at NuSirt.
Well, thank you for having me. I know you've already heard a little bit from NuSirt during the course of this meeting. I'm aware of the time, so I will get right to it. I want to focus. You heard a little bit from Joe Cook about our pharmaceutical applications. I want to focus on our consumer health applications. And so what you see here is our vision, which is we focus on prevention and management of chronic disease. And our focus is using leucine combinations with combined with well-characterized compounds, either pharmaceuticals or nutrients or food components that have established safety profiles such that the combination optimizes energy sensing via sirtuins. And our focus is on cardiometabolic disease. We've made a fair bit of clinical progress already, as shown on this slide. In addition to the 3 phase 2 clinical programs that were mentioned earlier in this conference, focused on diabetes, obesity, and NASH, we also have run additional clinical trials using natural product combinations with an eye towards consumer health applications, using leucine combinations in weight management, pre-diabetes and cardiometabolic risk. And then we have a fairly deep pipeline, preclinical pipeline of other leucine combinations, which have demonstrated preclinical benefit in across a range of cardiometabolic indications, as well as lifespan. We are supported by a very robust intellectual property estate, 25 US patents issued to date, number of more additional ones pending. Earliest expiration is 2032. And those, of course, have the corresponding international patent protection as well. The core of our platform is based on this figure, and that is we have shown that leucine, as well as its one of minor metabolite, hydroxymethylbutyrate, or HMB, are allosteric activators of SIRT1, shown here, as well as SIRT3, enabling a leftward shift of the NAD dose response curve. Simply put, what it means is it enables NAD levels that are rather minimal, that occur in the fed state, to still activate SIRT1 as though you were in the fasted state. So basically a caloric restriction mimetic. That in and of itself does not provide a therapeutic, but when we combine that with other compounds that either activate SIRT1 or sit somewhere along the sirtuin activation pathway, we get fairly robust effects. This slide summarizes our platform, leucine plus at least one other co-activating compound. In this case, those that either activate SIRT1 itself or 2 other key energy sensors that crosstalk with SIRT1, AMPK, and eNOS, having effects potentially on a broad range of age and Western diet-related disease. Now, there is a potential continuum of solutions, and Joe showed a similar slide to this on Monday, I believe it was, ranging from the unsupported noise in the nutraceutical space to the well-regulated pharmaceutical environment, and in between, and this is what I want to be talking about today, is a trusted and validated consumer health application that has solid clinical evidence along with solid mechanism of action data to support it. And our intellectual property really covers this full span. So I'm going to summarize, starting with two combinations for which we have clinical data. For both, we have managed to show preclinically very nice improvements in insulin sensitivity, inflammatory biomarkers, body weight, visceral fat, total fat, and in, again, animal model systems, lifespan extension. Unfortunately, I don't anticipate being able to conduct a study like that in humans, but I guess none of us will. Clinically, we've shown that one of them, leucine B6, in a series of clinical trials, augments weight loss during mild caloric restriction, causes a loss of fat even in the absence of caloric restriction, improves insulin sensitivity, and that another one, a leucine-resveratrol combination, is quite effective in increasing insulin sensitivity and glycemic control. As you'll see, we believe it has other effects, but those are the only two that have been validated clinically. Leucine B6, why B6? I've already explained why leucine, and that's shown here in the center column. Well, the coenzyme form of vitamin D6, pyridoxal phosphate, we showed some years ago, inhibits somewhat the entry of calcium into the fat cell, and we've shown quite extensively that adipocyte calcium is a strong controller of the rate-limiting enzymes, one of which is shown here, for fat synthesis, for lipogenesis in the fat cell. So in the presence of leucine, we increase mitochondrial biogenesis and essentially energy demand by skeletal muscle, while the B6 suppresses lipid accumulation and allows lipolysis to increase in the adipocyte. The combination then enables you might consider it a shift in energy from storage in adipose tissue to oxidation in skeletal muscle in support of new protein synthesis. Let's see if I can, there we go. So on this combination, we've had 3 clinical trials, 3 placebo-controlled double-blind trials. We are able to show a nice increase in fat oxidation evident within 7 days. Maybe it's sooner, but 7 days is our earliest measurement point, and sustained for 6 months, which is as long as we've been able to measure, our longest study. If we do not allow any weight loss, if we essentially clamp body weight, we still see about a 2 kilogram loss of body fat. But if we impose a very mild lifestyle modification program, we see about a 9% loss of body weight over 6 months. That's twice the amount of weight that is lost, actually more than twice the amount of weight, almost 3 times the amount of weight that is lost under placebo conditions. This is a pharmaceutical level of efficacy. If this was a drug, this would be sufficient to get an obesity claim. We get a doubling of fat loss, a doubling of the loss of waist circumference, and significant and rather substantial reductions in insulin resistance, oxidative stress, and that should say inflammatory stress, my apologies. With the leucine B6 combination, we have extended this, excuse me, we have extended this to companion animals. We contracted a trial out in fat dogs, and in the absence of caloric restriction, we're able to demonstrate, you can see the leucine B6 in the black bars, we were able to generate weight loss that was statistically not different from that with a commercial weight loss diet in dogs, the RD diet versus placebo in the center. And you see this for weight change, fat change, and change in body composition score. And so we've actually licensed this just very recently. It's just hit the market. You see a package there called Slim Paws. My Zoom screen is obscuring the package, but you can see it. That is currently our first and only license. And this is our only, thus far, attempt to extend our human work into the companion animal space. It's an odd case where the human served as the model for companion animal. Leucine-Resveratrol, we tackled Leucine-Resveratrol simply because of the promise of resveratrol, which has been simply put unmet clinically. There are a few studies that can show some effects of resveratrol. Most cannot. That is largely a bioavailability and pharmacokinetic issue. So we asked if the shift in the NAD dose response curve might enable the levels of resveratrol that can be produced, that can be achieved in circulation to be effective. Here you see a PET scan of fluorodeoxyglucose in mice, in a mouse with a control, and resveratrol by itself looked pretty much the same. Leucine-Resveratrol, as you can see, it lights up quite nicely. Overall, we reduce plasma insulin. We increase muscle glucose uptake by 60%. Thomas is saying I have 3 minutes. So let me very quickly say that we have shown that we have this translated to humans. We've only done pre-diabetes. So these are patients with impaired glucose tolerance. And you can see nice improvements in HOMA with both leucine HMB that should say Leucine-Resveratrol and HMB resveratrol. I will not walk you through these curves because of the time constraint. We have a number of additional preclinical products for which we only have animal proofs of concepts. That does include strong synergy with NAD donors such as nicotinamide riboside and nicotinamide mononucleotide. We believe that this will solve the lack of efficacy, which is seen in clinical trials thus far with these products. So our key takeaways are that we have a clinically validated consumer application ready to go in weight management. We've got a companion animal program as well, a platform technology with a number of additional nutraceutical combinations ready to come online, strongly protected, and I think I've stayed on time.
Michael, thank you very much. Just one question. I mean, there's a lot of very complex science and data that you walk through. If you were going to give, let's say, a 30-second elevator speech to someone who wasn't so well versed in the science as you are, how would you describe what you guys are doing?
What we are doing is we are taking existing drugs as well as existing food components and nutrients and combining them with a well-known amino acid in a novel way to produce cardiometabolic and weight effects safely.
Great. Excellent. Good. Well, we really appreciate that. And thank you for the excellent presentation, Michael.